let's begin. You know my background, and I will be now guiding you through what I have learned over last 20 years of working with Western blot technique. A little bit shortly about Agrisera. Uh, well, we are located quite far north. Therefore, our winter days are looking like this, with a little bit of northern lights. And as we have entered now to the light period, our summer nights are never dark. This is very special location, and it's not a polar circle yet. And here on these pictures, you can see our facility. We have antibody production farm in this beautiful landscape of northern Sweden. And we also have a facility where we are purifying the antibodies and shipping them worldwide. As you know, we have been established in 1985. Agrisera has 60 employees and is part of Olink Proteomics since year 2020. Olink is developing diagnostic and research platforms where thousands of proteins can be analyzed in as little as one blood drop. And this is done using antibodies produced at Agrisera facility. We are also ISO certified. And our products cover not only antibodies for plant and algal research, but we also offer a complete product set for any lab working in proteomics. So we have a collection of unique epitope tag antibodies, over 1,000 secondary antibodies, and detection reagents for both Western blot and ELISA. Our secondary antibodies are suitable for a range of different techniques. Each slide has also the links. So once you receive the PDF, you will be able also to visit different blog posts and read more information. Western blot technique you are using in the lab has been around for quite a while. Please think now, which year do you think that this technique has been first published? It is a technique which allows to visualize a specific protein in a complex mixture. And the sample can come from different sources. It can be bacterial, it can be mammalian tissue, it can be plant. And well, it has been around for quite a while, and was developed independently in different laboratories, but the publication first publication came from this laboratory. Why the name Western blot? You're probably familiar with other techniques. Southern blot is used for DNA. Northern blot is used for RNA. We have also Eastern blot for post-translational modifications. And Western blot technique is for detection and visualization of proteins. And the name comes from the fact that the laboratory was located on West Bank and therefore Western blot, which fits well with the other names. And the concept of this technique is connected with a certain sandwich. So just to remind you how the transfer stack is looking, where the aim is to obtain exact copy of proteins separated in a gel, which are transferred using electrical current to a membrane. So the transfer stack is when using wet transfer, it's looking in this way. So we are applying the current. We have a gel, polyacrylamide gel, where our proteins are separated based on molecular weight. And then we aim to obtain exact copy of that gel. So remember, the copy needs to be identical. So here it starts, I hope it will be visible for you that the transfer conditions need to be also optimized as a whole process for the best result with the antibody. And I will walk you through that. But at first, we will look into antibody structure. Just a reminder, the antibody is a protein, and the most important part is a Y-shaped molecule, and the most important part is the antigen binding domain, marked here by yellow. This is the part which differs between antibodies, and that's where the antibody is binding to a protein. And there are different parts. This part is located in so-called FAB region, but we have also FC region, a handle. That's the region which does not change. And FC comes from the from the fact that when this molecule was investigated, it was the solution was left in the fridge and crystallized. So fragment that crystal is crystallizing, that's the handle. And that part does not change. It's a constant region, which is used, for example, for purification of the antibodies with the affinity to protein A or G. And here I'm showing you the IgG from goat, mouse, or rabbit, which you're probably using in your daily Western blots. So please remember that changeable part, where the binding to antigen occurs, is located in so-called FAB region. We also have antibodies from other species. I'm just showing them to you here so that you can see the difference. They are smaller, and that part is different and smaller. And here the binding to the target occurs. These are antibodies from llamas, which are easier, which can easier penetrate, for example, in immunohistochemistry due to their smaller size. And antigen binding domain is absolutely crucial. And the epitope is the place where the binding to the target occurs. And just a reminder for you how the epitope is represented. Here you will see the linear epitope, which is created by subsequent amino acids. And the minimum number of amino acids antibody is binding to is 3, but it can be up to 15. And the linear, so-called linear epitopes are recognized by antibodies in techniques where we work with denatured and unfolded protein. So it will be Western blot immunohistochemistry where tissue is fixed. Please remember this because this is very important for you to understand and succeed with Western blots. Antibodies are also binding to so-called discontinuous epitopes. And as you see here, these are brought by protein fold. So distant amino acids are brought together and antibody can bind. And this is a discontinuous epitope, which is recognized by antibodies in native techniques like immunoprecipitation, Western blot in native condition, or CHIP. So antibodies are binding to 2 different types of epitopes. In Western blot, we are dealing with linear epitopes. And now also a short reminder about clonality. you're probably using antibodies which are poly or monoclonal. And here comes, in my opinion, a myth that monoclonal antibodies are better. And I will show you why I do not think that way. Poly means many. So polyclonal antibody will detect a pool or will bind to a pool of different epitopes. So as you saw on the previous slide, an epitope can be 3, but up to 15 amino acids long. And we have polyclonal antibodies from different species, here ordered in the order of how much of antibody we can get from each species. So these are the polyclonals. And monoclonal, mono means one. Monoclonal antibodies are binding to only one epitope, and they can be produced in these different species like mouse, rabbit, rat, or hen. And the epitope is 3 to 15 amino acids. So if the monoclonal antibody binds to 3 amino acids in protein structure, it is very specific. But if by some reason that epitope is not accessible, the antibody is not going to bind. Polyclonal antibodies are more versatile because they cover much more epitopes. Therefore, they will be often working with broader range of techniques. And here is also one important thing when it comes to antibody stability, that in case a monoclonal antibody is losing its stability, then the antibody will lose reactivity completely. And this can actually happen. During storage, some antibodies may be stored for only a couple of years, while others may last up to 20, 40 years. Polyclonal antibodies, when they lose their reactivity, we still deal here with a pool. So what you may observe is a gradual deterioration of a signal, which is a sign that the antibody is starting to be losing its reactivity. So monoclonal antibodies are often used in diagnostic kits, but not always. And Olink proteomics platforms are developed based on polyclonal antibodies and a proprietary technique. Also, to succeed with your Western blot, you need to have an antibody which is matching your species and your application. And I will explain to you what I mean with that. This is a very crucial step when choosing the antibody which is going to be used in your experiments. And this is very often overlooked. So we can have antibodies which are made to peptides, to short peptides with a very wide conservation. In Agrisera, we call them global antibodies. So as you see here, the antibody will bind to a specific sequence which is the same in all these species. This is possible, and especially for photosynthetic proteins, the antibodies will be covering a very broad species range. And they will be very specific, but they will be binding to peptides which are 7 to 15 amino acids long. And these peptides are chosen from protein sequence. So how do I choose the right antibody for my protein? Here you can see a sequence of a photosynthetic protein, and let's think about 3 different antibodies made to that protein. We have antibody made to N-terminus, of course, after signal peptide, and then to a sequence from inside of the protein and from the C-terminus. And here is the catch. If antibody is made to a very short sequence, in order for the antibody to work, that sequence needs to be found in your target protein. So if you are aligning protein sequences in between with each other, and you don't know the peptide which was used to elicit a given antibody, even an overall sequence homology may not be enough for the antibody to bind if a peptide is located outside that homology. So it is required a 70% linear sequence homology for the antibody to bind. So please keep this in mind. And if you receive an antibody from your colleague, or if you plan to purchase it from the company, first step of successful Western blot is to check that you have the right antibody. And do not assume anything, because there are databases, there are available protein sequences, they need to be full length, and you can always ask the company, because the antigen information is often not included on product information sheets. But at Agrisera, we will be always happy to help you and check if a peptide, which we have chosen for immunization, is also conserved in your target protein, and therefore the antibody will have so-called predicted reactivity. Confirmed reactivity is when it is confirmed experimentally, and predicted when it is based on sequence identity. So please remember, first step of your successful Western blot is to check that you have the correct antibody. Once we have been contacted by a customer who purchased 6 antibodies and sent protein sequences afterwards to check. And 4 antibodies worked, but 2 antibodies did not, because the antigen was not conserved. And that was actually confirmed with his experimental results. So if he has done that step before purchase, he would save money. And there is also one question which I am receiving very often regarding the antigen sequence. Can knowing that sequence help to determine cross-reactive bands? Well, not really. And the sequences which we are choosing to produce antibodies are absolutely unique. So they are not found in other proteins. So antipeptide antibodies will be monospecific. They will be binding to very unique protein sequences. So even if you are blasting that sequence towards the proteome, you may be misled, and you cannot determine identity of cross-reaction in that way. And now we are entering the Western blot principles. And here is just a reminder for you how that technique is, what is it based upon. So we have a target protein transferred to a membrane, and we can either have a sandwich of primary antibody towards your target and a secondary antibody which is labeled with dye or fluorochrome, which will enhance the signal and facilitate detection of your target protein. That's one way of detection. But there is also another way, which is shortening your Western blot protocol time, where primary antibody is directly labeled. And primary antibodies need to be affinity purified to be labeled, and they can be easily labeled with fluorochromes or enzymes. And this way of detection is recommended if you deal with abundant and medium abundancy targets. It's not recommended for low abundancy targets, as enhancement of secondary antibody is important. And the Western blot workflow you're familiar with, it's having quite many steps. Many things can go wrong. And today I will cover this part up to protein transfer to a membrane. And in the second part of this workshop, I will be discussing the rest with background, with primary antibody incubation, and secondary antibody choice and detections. So you're welcome also to join in May for the second part, where these steps are going to be covered, and I'm going to address questions which are related to that part. But today we focus on these steps. So theory is very good. It looks good on a protocol, but results which you can obtain can be very different. Here I assembled for you 4 examples where we have sent the same setup of reagent, so primary and secondary antibody, to 4 different laboratories, which were all working with Arabidopsis thaliana, with the plant. And here you can see how varied results can be obtained with the same primary antibody. One lab reported no signal. The other lab was having a lot of background. You couldn't see anything. And the 4th lab finally was able to return results. And of course, if you are working with uncharacterized antibody, specific mutants need to be included. This is absolutely a basic in antibody world. Each antibody needs to be characterized on mutants. Overexpressors are also okay, but knockouts are actually the best. But I know they are not always possible, of course, to obtain if a protein is vital for the cell. So here you can see how varied results we can obtain with one antibody. And the question is why? And my question to you is, how much do you know about the protein you aim to detect? Because your success with Western blot starts at the desk, at gathering information. You want to facilitate a binding of primary antibody to a target protein. And to do that, you need to know biochemical properties of your protein, including isoelectric point and its solubility. Isoelectric point of a protein will be important for adjusting pH of extraction buffer, because if you extract a protein with a buffer which has a pH at isoelectric point when protein has no charge, then the protein will not be soluble. Important thing is also expression level of your protein. Is it abundant or is it of low expression? Also organ and tissue localization and cellular localization in a specific compartment. This is especially important if you are dealing with protein of low abundance. Amount of a target protein in a tissue, you may wonder, how do I know that? Actually, there are databases, and I am using daily the PAX database, which is a protein abundance database for different species, including Drosophila, including plant species. We also have, for people working with plants, we have this wonderful database, which is collecting data of expression for different plant species. And as a rule of thumb, over these 40 years of antibody production, we have seen that if a protein is abundant in a tissue in less than 10 ppm, which you will see in that database, you can check it up for your target protein, then a specific protocol is recommended. Cellular fractionation and precipitation are important steps. Otherwise, there will be very difficult to visualize your target protein. So your problems may be coming not from the bad antibody, but from not having enough of your protein target on a membrane. So the antibody has no target to bind. And when it comes to extraction, sample quality is extremely important. For people working with plants, following this link, you will be able to download free images of these model species, which you can use in your publications and presentations. Extraction can be done in different ways. We have time-efficient methods like bead beaters, and we have time-consuming extraction methods, which are manual, mortar and pestle, and sonication with freeze and thaw cycles. All this has to be done in a very prompt manner. So if you are extracting multiple samples, I would go for a bead beater, because extracting 20 samples by hand is going to be a rather laborious process. And also the variation in quality of extraction will be increased with the time you are getting tired. So you need to adjust your extraction protocol depending upon which material you're dealing with, and it has to be done very promptly. Any protocol which is including lengthy incubations and it will be endangering your target protein for degradation. One of the questions which were received before this workshop was about extracting proteins from samples with high lipid content, like, for example, Drosophila. And here there are a little bit of tips what can be done. I would definitely search a little bit in the literature to compare different extraction protocols, but lipids can be removed from the sample. And here you have a little bit of advice. There was also a question about trypsin, which is used when working with adherent cells, which are attaching to the walls of the vials we are growing the cells in. So yes, trypsin will be endangering proteins for degradation, because that's an enzyme which is cutting the proteins. So it has to be removed by neutralization medium containing, for example, serum. So this was an important point, an important question which has been sent before the workshop. And also when working with plant material, it can also be rich in lipids. And here are a little bit of references what can be done. So it is definitely a very important point to consider what type of material you are extracting a protein from. And we are preparing an educational poster which is going to be focused on just protein extraction. For low abundancy proteins, as I mentioned, it will be important to get enough of the protein for the antibody to bind. And in such case, I would strongly recommend fractionation. Please see here for yourself. Total sample, so like total leaf, for example, or total root, is not giving any signal. And once the fractionation is done, the target protein is concentrated, and finally the detection is facilitated. I'm certain that quite many antibodies were discarded or abandoned when total extract was analyzed. For low abundancy proteins, it is a must to go to specific cellular fraction. You may ask me what to do if protein is cytoplasmic. In that case, one can precipitate. So, for example, do TCA acetone protein precipitation, and that way load on the well as much as possible. So, like 50 microgram of protein and more for low abundancy proteins. And the next step, of course, which is important to consider, is extraction buffer. How your extraction buffer, how many compounds there are. Because you can see here, on this example, there was a test lab which was using tree-spaced buffer and hippie-spaced buffer. And just by changing extraction buffer, target protein was visualized. In some cases, addition of strong denaturants like urea may help to unfold the protein and facilitate antibody binding. In this specific example, we have so-called garden blot. So we have samples from barley, Arabidopsis, and rice. And the sequence have been checked, which was used to elicit this antibody, and it was perfectly conserved in all 3 species. But no reaction for 2 of them. After addition of urea and keeping the protein unfolded, the antibodies were having places to bind. So buffer composition is an important parameter to succeed with your Western blot. We also need, in many cases, we need to use specific organs. So it can be a 2 millimeter root tip, for example, or a specific developmental stage. You need to analyze this before you go to the lab. We want to, of course, include protease inhibitors, in some cases proteasome inhibitors, if your target protein can be degraded by proteasome. There was a case of a lab which was trying to detect a protein for around 1 year, and there was empty blots all the time. And then finally they added proteasome inhibitor and the band came up. You need to also work in optimized environment and obtain a good yield of protein extraction. So highly diluted samples need to be concentrated, and you need to have a proper amount of buffer in relation to sample. And good protein concentration will allow you to load more per well and therefore increase amount of your target protein, especially if it is low abundance. How do we work? How should we work in the lab? I wonder if you're familiar with that shortening, GLP. What does that mean? Think for yourself. Good laboratory practice. It's an absolute base to succeed with Western blots. So we are working clean. We're cleaning the equipment between experiments. Absolute must is to conduct gel electrophoresis and Western blotting in the cold with ice-cold buffers. And it was one question which was sent here about bands which were bent, and that's a result of not running the procedure in the cold. We need to be precise. When did you calibrate your pipettes last time? It should be done at least once a year. And we need to use fresh reagent stock. We do not reuse the buffers. And we follow the protocol, which means that we have a chance to obtain consistent results. Gel electrophoresis, that was a question I received about different types of gels which are available and do they have any influence on protein separation. As you see here, yes, they may have an influence and increase the detection range. In some laboratories, we are using precast gels. So, for example, at Agrisera, we are using Novex gels, which are precast because we want to have reproducible results. Shape of the bands, if you're casting gels yourself, can be sometimes compromised, and the polymerization between different gels may not be exactly the same. It is influenced by the temperature. So precast gels will give you a chance to have more controlled and reproducible conditions. What should I load per well? That's also an important thing, therefore I have included it. Protein chlorophyll for photosynthetic tissues or volume. And definitely recommended is microgram of protein or microgram of chlorophyll. I'm not recommending to load based on volume, especially if we are doing comparisons or quantification using Western blot. I often receive the comment that, well, it's not possible to measure protein, estimate protein concentration in extraction and loading buffer. And actually, this is wrong. There are reagents like this one, which are compatible with detergents, with salts and metal ions. So you can estimate protein concentration in the presence of these compounds. And please remember that when you are extracting proteins, you need to work fast. You need to be very vigilant and follow the protocol and be efficient and complete the extraction in a very short period of time to avoid protein degradation. One of the questions which I received was how much material should be loaded, should be microgram or nanograms. And it depends what you are loading. If you're loading recombinant protein, of course, it's going to be nanograms. And if you are working with an extract where your target protein is of low abundance, then the load needs to be increased. And here you can see titration, which was done. So varied amounts of protein per well were resulting in less background. This is a step which can be done before, for example, if you would like to obtain a good, nice figure for publication. As you see here, the background became less intensive when antibody was used in this high dilution. All these parameters are very specific to protein antibody pair, and they need to be worked out. Another question which I received was about using one-dimension or two-dimension gel electrophoresis. You are probably familiar mostly with one-dimension gel electrophoresis, which has its benefits. And what you need to keep in mind is that in one dimension, proteins are separated in a polyacrylamide, which is like a net with different holes. So proteins are separated based on how do they migrate. So heaviest are getting stuck on the top of the gel, and the smallest are moving down the gel. And in some cases, if you're working with proteins of very low molecular weight and you run your gel electrophoresis too long, they may actually get out of the gel, and therefore you do not have a signal on your blot because the target protein left the gel. And each such band will be comprising of hundreds of proteins. And in two-dimensional gel electrophoresis, we are separating these bands into the spots, specific spots. It's possible to conduct Western blot also on 2D gels, but results will be looking a little bit different. It will be spots, not bands. Sample denaturation. Please answer in your head, in what temperature are you conducting sample denaturation? Probably this one. There are different variations of this protocol. In our work, we are using 70 degrees for 5 minutes because excessive heating of proteins can lead to aggregation, like in case of H+ ATPase, which will aggregate when boiled, and therefore no band on the blot will be detected because target protein was still stuck at the top of the well as an aggregate. Protein transfer conditions. This is how we are doing it in ice bucket with ice chilled buffers. And here it is a very important thing to consider. Is it better to run semi-dry or wet transfer in a cold room to avoid overheating of the instrument? Yes, definitely in cold conditions, but you need to take into account also molecular weight of a protein you're aiming to detect. Because Smaller proteins will be more easily migrating from the gel to the membrane. So transfer time will be shorter for proteins below 20 kilodaltons, while proteins of higher molecular weight may require longer transfer time. And also, please see the difference. You can use different pore of the transfer membrane. High molecular weight proteins require more SDS, while smaller molecular weight proteins may require more methanol. In general, for high molecular weight proteins, wet transfer tank is recommended. But of course, semi-dry transfer with well-optimized protocol may work well as well. But before you go to the lab and you start your gel electrophoresis and transfer, please look at that guide and consider your transfer conditions. Because if you do not have enough of your target protein on the membrane, then the antibody has no protein to bind to. And that can be a reason to low signal to noise ratio and lack of successful detection. Protein transfer, which membrane to use? Please answer for yourself if you're using PVDF or nitrocellulose. I'm not saying that one of these membranes is better than the other. They have different properties. So PVDF is protein binding is based on hydrophobic interaction. PVDF can be air dried and re-wetted, and that's an advantage because it can be stored for longer time, up to a few months. It has better physical strength and chemical compatibility. Nitrocellulose has another mechanism of protein binding and is also more fragile. And I would recommend to have both membranes in the lab. And one protein antibody pair can give better results on PVDF, the other one on nitrocellulose. So there are always exceptions. And oftentimes on our product info sheets, we are informing if it is preferred to use, for example, nitrocellulose. Protein transfer type. Which one are you using? Are you using wet transfer or semi-dry transfer? There are differences between them. We are using more buffer for wet transfer. It's another electrode contact also in wet transfer, and the speed is different. And it is semi-dry transfer is more maybe more difficult. Therefore, for beginners, wet transfers are recommended. I'm not saying that one of them is better than the other. If it can work better for certain types of the proteins, and this needs to be simply worked out in the lab. How to efficiently remove air bubbles from the wet sandwich. So please do not do like that. If you would like to obtain sharp bands, assemble the sandwich directly in the cassette. And on this video, you will see how to push out gently the air bubbles. And do not squeeze the gel and do not do drastic things to it. You have to be very gentle. And the sandwich needs to be tight for the best contact between membrane and gel and for successful transfer. Quality control of your transfer can be done by reversible stains with Ponceau or Kumasi R250. And why are we doing it? We are doing it to see if a gel is looking like that. I mean, the transferred proteins are looking like that, then we will not continue with that membrane. Probably it was too hot, it was too much of a protein loaded, so this experiment is wasted. The stained membrane after transfer should look like that, and then we can proceed to further steps. A good control to do to check the efficiency of your protein transfer is to actually stain the gel after transfer with Kumasi. So once you disassemble the sandwich, it's very quickly done. You stain the gel in Kumasi. And if there are any proteins left, it's no good because it means that your transfer conditions were not good enough. Here we assembled 2 gels in the sandwich. And as you see here, the second gel is not looking good. It should be also transparent. There are proteins left behind here. And therefore, we decided for quantitative Western blot to always use 1 gel in a sandwich, not 2. So staining the gel after you disassemble the sandwich may help you to understand if your transfer has been done correctly. If all proteins in the required molecular weight range have left the gel and migrated to the membrane. Please remember what I mentioned about low molecular weight proteins, which if you apply too long transfer conditions, I mean, you continue the transfer for too long, you can blast low molecular weight protein from the gel through the membrane. And the way to check that is to place 2 membranes in the sandwich. Because if you stain both of them with Ponceau and you will see the protein on the membrane number 2, it means that you blasted it through. So as you see here, lack of detection by antibody can be actually due to lack of a protein on the membrane. And now I am shortly going to mention issues with antibody format. Why is it important to consider? Because primary antibody, which you are using, can be provided as serum. It means it's not purified. It can be purified to total immunoglobulins, and it can be antigen purified on a specific protein or peptide. And when you are purchasing antibodies, please pay close attention to it, because if you are purchasing antibody present in total immunoglobulin, it may look like a lot because it may be 0.5 milligram. But that 0.5 milligram may not contain so much of specific antibody. So, oh, apology, I was too quick here. So here, amount of specific antibody in serum cannot be determined unless the serum is purified. When you purchase total immunoglobulin antibody purified on protein A or G to total immunoglobulin, your specific antibody will be only between 0.5 to 5%. Most suitable for applications like CHIP, but also Western blotting of low abundancy proteins are antigen purified antibodies. And just a small reminder how purification is done. Here you can see our lab. We are using affinity purification with columns where we have been binding the antigen. So the peptide is bound to the column, and it can also be protein for antibodies developed to proteins. And then we are passing a serum through the column. Specific antibody will bind. And then by applying usually low pH, we are disrupting the binding between the antigen and antibody, and we elute the specific antibody which is collected. And here, this is a very exciting question. Antibodies are expensive, and many labs are reusing them. Maybe you are doing it too. And how many times can primary antibody be reused? What do you think? 1 time? Some antibodies can be reused 20 times. So the buffer containing primary antibody is collected and stored in a freezer, thawed, and the antibody some antibodies can be reused quite many times. However, not all of them. So you may have encountered lack of a signal in a second incubation. And this means that your antibody can be used only one time because there's all primary antibody bound in this one incubation. So there's nothing left. So there's nothing wrong with the antibody. It's simply that it is enough of it only for one blot. So this has to be experimentally determined for each antibody. Please remember, antibodies are like people. Everybody is different. So each protein antibody pair is different. And here it is also an important point which has been, which I was asked about, that sometimes antibodies accidentally stop working. You maybe also encountered this in your work, and there can be different reasons for it. It can be freezing and thawing, or your freezer broke down. It can depend upon addition of stabilizing substances. And also, if you're reusing an antibody multiple times, of course, at certain point, it will not work any longer. Important point here is that it's not possible to predict antibody stability over time because it's determined by this antigen binding domain. The place where antibody is binding to the antigen may influence the stability of a whole molecule. So some antibodies may be working for 20 years, the others may lose activity after 1 to 5 years. So please keep this in mind and store your antibodies properly. Read product information sheets because you cannot assume that all antibodies require glycerol. There is no need to add glycerol to serum, for example. So you need to be really careful and check the information on the vial and on the product information sheets. If you have an unknown antibody, you can do a test and store it in different conditions. Usually when it comes to temperature, minus 20 degrees is quite enough for storage during several years. Some antibodies may be more stable when they are not frozen. Therefore, you have to be careful and check product information sheet if you're buying catalog antibody. And now we are going towards the end. And I would like to introduce to you our offer of over 100 free antibodies. New antibodies are added every week. There is a pool of antibodies to epitope tag, to different bacterial proteins. We are providing free primary antibodies, secondary, and ECL, and in return for results within 3 months. Here's a link to that offer where you will find the antibody list. So this is now question time.
Yes, there's two questions for now. One is in Western pictures. Sometimes van will be connected from left to right. What situation can cause this, and how can it be resolved? And there's a picture in the chat. If you can open it and look at it, it's like a black bands connected from left to right.
Mm. Is this? I wonder if it looks like it's too high protein load. There is too high protein load, and therefore the wells are stacked together, I think. And I wonder also if this is a precast gel, or if it is a gel which was casted by a researcher, because that can also influence it. Are there any more questions? It was very good to paste the picture. Can you explain adding C on terminal of peptide to antigen to get antibody, and please explain conjugate ovarian? Okay, a cysteine is added to C or N terminal of a peptide to facilitate its conjugation to a carrier. Because peptides are very small, and for immune system of the host in which we're producing the antibody, to enable the peptides to be seen, they need to be stuck on something bigger. And KLH is a protein from mollusk, which is huge, 1 million Daltons. So using chemistry with we are adding the seed at the terminal of the peptide to facilitate its conjugation to a carrier. So, of course, during the antibody production, there will be antibodies produced to our target, but also, of course, to a carrier. And ovalbumin, we are not using it as a carrier. It's much smaller. We are using mainly KLH or BSA, but we prefer KLH as it's more immunogenic compared to BSA or ovalbumin.
Okay, Dr. Joanna, for the previous question, in Western picture, sometimes veins will be connected from left to right. It's because it seems like in the picture, it seems like overloading. Am I right?
Yes, that's what I'm thinking.
Okay. Okay. So what's the situation that caused this is because it's overloading. Okay.
Yes, and I
So
hope you are measuring protein load per well. So when you overload the gel, you can, and also when you, I think that one of the reasons can be that you should have been taking pictures a little bit sooner, because it may be also that you are using too sensitive ECL. So the picture is coming, the bands are coming extremely fast. So if you decrease the sensitivity of ECL, it may take longer time for bands to come, and then you will record them when they are separate from each other. Actually, if you participate in our quiz, you will get a chance to win 2 ECL reagents, which you can use in the same experiment. So if you use 2 sensitive reagent, you can wash the blot and apply the other ECL. So you can save your experiment by playing with 2 different sensitivities of ECL.
Okay, so you can wash the blot by playing.
Yeah, you can wash the blot with your ordinary wash buffer and then apply ECL with lower sensitivity.
Okay.
Yeah, hello, Professor. Yeah, may I ask one question?
Yes, absolutely.
Yeah, so actually, I'd like to ask that, for example, if I added one antibody to the membrane and see my protein band, and next day, if I want to use another antibody, can I use the stripping buffer, or it's not a good idea?
Well, in the second part of my workshop, I will show you how you can use simultaneously 2 primary antibodies on the same membrane. But they need to recognize target proteins, which are 20 to 30 kilodaltons apart. Stripping of the membrane, you have to keep in mind that you can also strip your target protein. So it is rather harsh treatment. So if you deal with high abundancy protein, that may not be a problem. But if your target is of low abundancy, you may actually wash it away remove it from the membrane while stripping and increase background.
Okay, thank you very much.
You're welcome.
Okay, there's another question from Zoe Pan. Is plant rubisco and alpha tubulin proteins have similar molecular size, making it difficult to detect tubulin? How to improve this situation?
It depends if you need to detect both proteins, or if you could use another. If you are using alpha tubulin as loading control, you could use another protein of another molecular weight as loading control, which would be further apart from Rubisco. But yeah, it's you can also play with percentage of the gel, which would then allow to separate these proteins a little bit better. But you can also check here for loading controls of other molecular weight. Please keep in mind also that there are no universal loading controls, because even actin and tubulin, their levels will change depending upon your experimental conditions. I see other questions. How do I know prevent smiling or frowning blots? Well, low temperature. You need to conduct your gel electrophoresis and Western blot in cold and with ice cold buffers. Why is it that some antibodies can cause spotting on blots? For example, collagen F. Uh, well, I would think that maybe blocking is not enough. So at least when working with plant material, we know that BSA is not so efficient as a blocker. So you may try another blocker. I am going to address a blocking issue in the second part of this webinar in May. There are also commercial blockers which one can test. Thank you very much and good luck with your blots. And I hope you join on the second part in May.
Okay, hope to see you all on our second seminar workshop. Thank you.
Thank you.